Okay, before we continue with lecture number 3, someone asked a question in through the WhatsApp. Okay, and then I have copied that question and a response to that question into the forum. So you can always go back to the forum and have a look at the response that I put there, in case you can learn something from the question. All right. Um, it is about the previous lecture where am I sharing the slide now?
No, bro.
Oh dear, sorry. So sorry about that. Drawbot. Okay. All right. So someone asked question related to detecting the polymorphism. Okay, polymorphism, as I mentioned, the general idea is that uh, on the same position of the gene, okay, for example, any gene on the chromosome, all right, with this region, there is a difference. For example, this one is chromosome 1. So a pair of chromosome, isn't it? Everyone has a pair of each chromosome number. So pair of chromosome 1. There are something different over here that is slightly different between the homologous chromosome. So when you have that, you can have a polymorphism as well. And then in another individual, if you are same chromosome at the same position relative to this, also similar changes. So polymorphism among individuals, that one can also call polymorphism or variation. Okay, so the term polymorphism variation you will come across quite often when you talk about population. You know, many people, and then we are talking about uh, same locus, might be slightly different between one individual to another. So you can have a variation, and you can also call it a polymorphism. And polymorphism can be because of the changes because of the differences in terms of the repetitive number. If you recall the microsatellite, the mini satellite, and even at the single nucleotide polymorphism, whereby the difference is only between uh, that particular position between AT and GC. So that will be polymorphism, okay, and variation. So, and how we detect, for example, certain locus, we can detect when we cut and the other one cannot be cut. So when we want to cut the DNA, then what we do, we use restriction enzyme, right? So we use a restriction enzyme to cut the DNA. And restriction enzyme, I think you have learned somewhere else also that restriction enzyme is basically a molecular scissors. Okay, so molecular scissors in which it is an enzyme, many different enzymes that belong to the same group. We call it restriction enzymes. So these enzymes are restriction endonuclease enzyme. They recognize different nucleotide sequence. For example, like in this case, for from the microbial of Thermus aquaticus, so T and then AQ, so TEC. So TEC1, the Thermus aquaticus, it recognizes four recognition sequence, whereas ECO R1 from E. coli, okay, ECO. Usually the nomenclature of the enzyme is based on originally where they were initially isolated. Okay, like BAM from Bacillus amylo liquefaction. So BAM, 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 and then H and then variant number 1, for example. So it is always like that. The first three letters indicated genus and also the species, okay, genus and species. So all nomenclature will be like that. And then the D3 number one like that, H1. It is based on the variant of the collection that they have when they initially went to the field to collection, so they indicate as this number. So you can see that six cutter, you can have six, you can have eight, and then you can have much longer with a different name. But all of them, the concept is more or less the same, whereby they are called palindrome. So if you read from 5 prime to 3 prime, so like this case, you read GA, T, C, right? And then you read the other way on their uh, corresponding strand, complementary strand from 5 prime to 3 prime. So complementary strand, we also read from 5 prime to 3 prime. So it will be G, this one will be A, this one T, this one C. So you can see the reading from 5 to 3 on both strand will be the same, GA, TC. Same, if you look, all of them, they are the same. So these are called palindromic. Okay, palindromic, which is actually from derived from the word palindrome. Okay, from the word palindrome. Okay, palindrome. So it is palindromic characteristic whereby you read from the one side, you read from the other side. It is the same, just like the number 2992. You read from here and then you read from here. They are same reading. Okay, for example, you can search for in the internet not only for restriction and nucleus, even words can also, or sentence can also be uh, palindromic. Okay, like able, able, and then Elba. Uh, is an island or a brand name for your jeans or your t-shirt or whatever. Okay, so palindromic. So all restriction characteristic, yeah, I saw someone wrote Riska, yeah. But, so that is palindromic. So all restriction enzyme are, what do you call that, palindromic. I'm trying to have the chat open so that I can. Okay, do we need to memorize where the site the restriction enzyme will cut? You don't need. You only need to understand the concept. What is restriction enzyme? And if you can like remember one or two example, like the common one. Common one is like eco R1, and then what else? Hindi 3. So these are usually common restriction enzyme. Eco R1, BAM H1, Hindi 3. So these are common one. You can, if you can recall the sequence, it is fine. Okay, but not necessarily that you have to memorize this. I don't uh, go by that requirement, right? Probably it's good later on, when you are doing any analysis of the DNA when you are in final year project, or when you are masters or PhD, you are working with the enzyme and then you are looking at the sequence, and then you notice, oh, this GAA TTC is, oh, this is a eco R1 restriction site, then it's okay. But for now, you just remember the concept of restriction enzyme, where this the place where the enzyme can recognize a specific location on the DNA, and it can cut it over there. All right.
Doctor, you're muted.
I don't know when did I press the mute button. Okay, you recall the chromosomes, and then you learn about they are compacted. If they are uncompact, then you can imagine all the restriction enzyme converge and trying to find the position to cut the the DNA itself, okay, the double-stranded DNA, not the single, the double-stranded DNA. So that is the function of the enzyme. But later on, probably you might want to ask, then how come the restriction enzyme doesn't cut the DNA in its own cell, right? So you might want to ask that. So that one in another topic, but they are a way to protect. For example, I have a scissors, I can protect myself from being cut by my own scissors, for example. So that's the concept behind restriction enzyme not cutting its own DNA. Okay. Uh, all right, and then based on the polymorphism just now, earlier, for example, single nucleotide polymorphism. So on the chromosome, we this one is double-stranded DNA, right? So we have a pair of chromosome, which is the double-stranded DNA, and it is so this double-stranded DNA is represented by a single line like this. But when we uh, uh, magnify this position, actually it's a double-stranded, so it looks like this. Another chromosome at the same locus, remember the locus, the location, the locus, also double-stranded like this. And then this area is represented only by a single line over here, but in actual fact, this one is also a double-stranded DNA, okay? So, but we are focusing on these areas like this. So when you see like this, in the chromosome of one individual, you can see in some individual, at this sequence, it has the restriction site. Okay, restriction site, CCT, CCT, CCTGAGG. Okay, so this will be one restriction site. Okay, and then on another chromosome, actually there is a difference at this position, for example. Okay, so when that happen, on this chromosome got restriction site, this chromosome don't have the restriction site. So if I use the restriction site to cut this enzyme, this MST2 enzyme, then when the enzyme comes in, okay, enzymes comes in looking at the chromosome, at the DNA strand, and then it can recognize this one, but it cannot recognize this one. So this enzyme cannot go over here, but can go over here. So when that happens, this can be cut, right? This can be cut. The DNA will, if like this, DNA can be cut. Whereas this one cannot be cut by MST2 enzyme because the recognition sequence of this MST2 enzyme is for CCT-GAGG, but this one is CCT-GTGG, so it cannot be cut. So this the enzyme can excise over here, but this one cannot excise, cannot cut, right? So when that happens, so you just imagine if I have this enzyme and I want to analyze, do I need to analyze the entire chromosome? I do not need to do that, isn't it? So how do I just only want to concentrate on this area only? So the way to do it is by make a copy of this chromosome. So I can make a how do you make a copy? You learn in the previous slides of this lecture, you do a PCR. So when you do a PCR, then you need to form a primer here and then a primer here. Same because this sequence are homologous chromosome, then the same primer can be used here. It will also recognize this position. So you amplify this region only. Okay. So this is what is meant by left primer, right primer. So you only amplify this region, okay, this region. So when you amplify this region, when you have a PCR, then the product will be only the sequence, the fragment of this area only, isn't it? Many, 1 million copy. And then this one also, same primer, it will also copy a 1 million copy of the same sequence from this chromosome. This one, this, okay. So now, after I treat with the enzyme over here, MST2, then the enzyme will be able to cut all this. Whereas this enzyme, it cannot recognize this position, so they remain as a single-stranded, many strands of a single strand amplification of this region, right? So you see, and then you transfer the information that you have of here into here. So what happened in in this DNA that has been amplified? When you cut with the enzyme, then you have how many fragments here? You have two fragments for this one. This is one fragment. This is another fragment, isn't it? Whereas for this one, because the enzyme cannot cut over here, so you have one fragment only. One fragment here means that all fragments are the same, actually a copy of the same DNA that was amplified here, isn't it? So based on this, when you run the DNA on the electrophoresis, so this one is the electrophoresis gel, so like this one, right? The gel like this, the gel like this. Then you put the sample in the well over here, and then you put an electrical charge, then it will migrate. So the shortest one will move first. Okay, by the way, the shortest one will move first, or the longest one will move first? Will move faster. Which one? Jet, shortest. Okay, someone. What about the others? Shortest or longest will move faster? Shorter, faster. Right, good. So yeah, all right. So it's very easy. You know, you are like in a crowd, and then you are carrying very big spoke, and then you just walk without anything. So the one you will move faster compared to your friend carrying a very big box. It means it move slower. So like so same like this. So in this case, what you will then see is that this fragment, the 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 short the short fragment of this, then will move further to the bottom. Whereas this one will move a bit slower, then this will be the second one. So this one is number one, this one number two, this one number three. So number one, number two, and then number three, right? So this is an example of how the migration of the DNA. And then what about this? What about this? And then this one is the same like this one. So what is meant here is that based on the analysis of the DNA, actually this one is from individual, hang on, yeah, sorry. This one is from individual number one, individual number two, the father and the mother, and then the children, individual number three, individual number four. No, not in the sequence. I don't like it. Okay, we redraw it. Okay, I do it this one number two, this one number three, this one number four. One and four is father and mother, and then this one is the children, brother and sister. So what happened here is that this father, when you take his chromosome, the one that I drawn here earlier, and then you do a PCR like this, and after the PCR you treat with the enzyme, and then you take the father's DNA that was amplified and run. It was noticed that this one is three bands. So three bands mean that for the father's chromosome, the father is heterozygous because one of the chromosome have a restriction site, so forming two fragments consisting of this, and then the longer the longer fragment actually is amplified from the other chromosome of the homologs from the father, so the third one. So the father is heterozygous, and then the mother is also heterozygous because you can say one, two, three using the same concept, one, two, three, one, two, and the third one. So the three chromosome, three fragments, in which this one explaining the one that is not cut, and this is actually from here and also from here. So you can see this from father, from mother, and then when the father married the mother, have the children, and then the children also done the same PCR, take his chromosome, take his DNA, run the PCR, amplify, and then for the children, when run on the electrophoresis, then you find out that, oh, only the long DNA, the long fragment is present. Means that for this boy, the fragment is actually, both of them are, um, okay, both of them are consisting of a long fragment. Okay, long fragment, this one also like this. Meaning to say that this boy do not have the restriction site over here. So the boy has a polymorphism in which the boy also have same sequence like this on his other DNA. So that is why you have only one band. Okay, why is it one band over here when you have this, and this? Why one band? Why is it only one band? This and this. Yeah, it's not cut, but this one although they are from different DNA, amplified from the different DNA, the concept of the DNA when you run on the electrophoresis is based on the length. Okay, so even if I have in another story from chromosome 1, 500 base pair, and then from chromosome number 5, I run also 500. If I run on an electrophoresis gel, I will only have one band because the DNA electrophoresis is based on the length. So electrophoresis cannot tell 500 from chromosome 1 and 500 from chromosome 2 in different other story, right? So that is the concept behind electrophoresis. So for this boy, you can see both of his amplified DNA of the chromosome have only one DNA strand in which the boy do not have the restriction site over here, so DNA enzyme cannot cut. So like this. And for the girl, it is homozygous for the one with the restriction enzyme. So the brother do not have restriction site. The sister homozygous for uh, this area in which the sister have the restriction site for both of her chromosome. Okay, so that is why you can see like this. So this is the concept to differentiate between one individual to another based on the presence or absence of a difference in one nucleotide. Like this one is GC, this one is AT or TA. Okay, so this is how you do it. So you example of this were you because in the sickle cell anemia, some of the sequence the difference is only at one nucleotide, causing the individual to become sickle anemic individual, okay? And then we detect by the PCR. So this is to explain, very long explanation, almost half an hour, oh my God. Okay, so this is how it was, how to explain all this. All right, so coming back to your, so I'm going to stop sharing for a short while because we finished with this one. So DNA. So, doctor, so the question is AA. What is AA here? I don't understand that question. Elaborate a little bit while I try to get the other. AA means if you are talking about homozygous, yes, it is homozygous. So those both chromosome are the same, so they are homozygous. In your case, probably you are meaning that AA is a homozygous for both chromosome are the same, so they are big A, okay? Or if they are recessive, a small a. Right, so that basis of homozygous to cause the disease and sometimes heterozygous can also cause disease is also one of the way certain disease are shown in human. All right. So I'm going to share the next slide. Genetically based. enzyme deficiency in human. So this one is right. So genetically based enzyme deficiency in human. Uh, you imagine like you have learned in Mendelian genetic, uh, big A cause red color flower. Big A, small a still uh, red flower because the big A is dominant, right? But then when there is an small a, it becomes homozygous recessive, then it becomes white, right? So that concept can be used in certain diseases or deficiency that cause disorder or disease. Actually disorder rather than disease, okay? But sometimes I use it interchangeably. So this big A or and then big A small a show a normal. Normal means red. Imagine the Mendelian genetic red, whereas small a is a homozygous recessive and it caused the disorder or disease just now. So that concept, there are many diseases or deficiency or disorder in human is caused by the single gene. Okay, so in this case, there are many examples like this. So these are the genetic defect that is described uh, or the disorder, and this one is the enzyme that is mutated, and then because of the changes in this enzyme, it caused the disorder. And this one is the position or the locus on which chromosome in human that have this kind of changes that cause this disorder. Okay, so I hope you can still recall what is meant by 3Q or 7Q or 17Q2. Okay, so I hope you can still recall what they mean by that. Okay, uh, what actually they mean by that? The 17Q or whatever. 17Q, 1P. The yes, okay, the long
The
arm shot.
long arm, short arm, yeah.
Yeah, good. Okay, long arm, short arm. So this one is the position, the number, the first number is the chromosome number. Okay, and then long arm or short arm. So these are example, and usually, there is a database by John Hopkins University that collect the information of this um, disorder in human that is caused by some changes in the DNA that produce these enzymes. Okay, so it is OMIM, online Mendelian inheritance in men. Okay, in men. Men means in human. Okay, so these are some of the deficiencies of the enzyme that cause disorder by a single mutation, similar like the Mendelian concept of homozygous and dominant. Okay, and then some mutations create a simple phenotype, while others are pleiotropic, meaning to say that some of this enzyme, when there is a mutation of this enzyme, and this enzyme is not become abnormal enzyme, then the symptom or the characteristic of individual having the defective enzyme is the same. So simple phenotype. For example, the the phenotype shows that the hair becomes red color naturally. Not red color. Red color is common in human. Somehow the hair become blue. No, there is no natural blue hair in humans, isn't it? But somehow because of the mutation, it called a simple phenotype of blue color. So you know that if that individual have blue hair, then it because of mutation of this enzyme. Hypothetical situation, okay? So, but in some other uh, deficiency, although the individual have, for example, not a specific phenylketonuria, but if let's say I use this enzyme in individual number one, it has a symptom of only the lung problem. In individual number two, it has a symptom in the kidney only, but the lung is normal. Okay, whereas this one lung problem, kidney normal. This one kidney problem, lung normal. So meaning to say that between individuals, although they have the same deficiency, the characteristic of the disorder in different individuals are different. So that is what is meant by pleiotropic. Okay, so you cannot pinpoint specific disorder characteristic based on the changes, right? And then this one is another example, similar like this. Okay, another table, example of human diseases, mode of inheritance, and associated genes. So associated genes are usually related to the enzyme, okay, or sometimes related to protein. Okay, so this one is protein. This one is the enzyme. This one is the protein. Uh, this one dimension. Okay, most of them are protein. This one is the enzyme. Then this one is a protein. So you can see from this, uh, these are changes on one position in the human chromosome. So it causes the disease or the disorder like this. And then you can see type of inheritance. What is autosomal? Autosomal means what? Autosomal and then X-linked. What is autosomal? Autosome. What is autosome? Hmm? Anyone? I'm looking at the chat, but nobody. Non-sex. All right. X-linked related to sex chromosome. So the autosome is the one that is not the relate the is the autosomal chromosome, which is not the X or Y's. Okay. So when you see this, you can see that uh, not somatic cell. In the somatic cell, we also have the X and Y chromosome, isn't it? But uh, but this one is talking about the chromosome itself. Okay, someone answered somatic cell. I just want to clarify that as well. Okay, so in the disease that is caused by recessive, what is meant by that? If a disease is caused by a recessive, it means that both chromosome has to be in a homozygous form in order to show symptom. Okay, for example, like this one, two chromosomes, chromosome one. This one is big A. This one is small a. Okay, so small a is the one that has changes that can cause the disease. But when this small a present together with the big A, you write like this. Will this cause a disease? It will not cause a disease, right? Because this A is uh, producing the normal enzyme, and then in this A will not the cell, our cells tissue will use the enzyme produced by this chromosome, whereas this one will not be used because it is defective. Okay, so in order to cause the disease, then you can have another individual that has the a small a as well, then this individual is then will have small a. So now both of them have the defective chromosome, okay, and then because of that, there is no big A present, isn't it? So when that happens, there is no big A to compensate for the absence of the enzyme from the big A. So only the enzymes, the defective enzyme is produced in this individual, then only this individual will show the symptom, the disorder or the disease. Okay. So when you come across recessive disease, then straight away you have to remember that they have to be in a homozygous form in order to cause the disease. Okay. And then if they are in the heterozygous, individual one, individual two, individual number 1000. If they are heterozygous, they will be a normal individual because this big A is producing the normal enzyme to cover for the inability of small a. But in another individual with small a, then this will be the abnormal individual. Okay, so when you come across a gene that cause the disease in the form of autosomal recessive, or sometimes they mention as recessive only, straight away you have to know that they have to be in a homozygous recessive. Okay, it cannot be in heterozygous. But then you come to dominant then. Dominant. So dominant is another case. So in a dominant disease, okay, let's say this one chromosome number 2, okay, if this one is big B, this one is small b. So you have, and then another individual with small b, and then another individual with big B. So out of this individual number 1, individual number 2, individual number 3, which one will uh, have the disease on those with the autosomal dominant disease? Okay, think of one number, 1, 2, or 3. Which one will have the disease if the disease is autosomal dominant? Okay, so 1 and 3. All right. Okay. So I think almost all of you answered 1 and 3, which is uh, 1 and 3. You are all correct because only one copy can straight away cause the disease because it is a dominant, right? Dominant disease. So that is why it's called autosomal dominant. So only one copy is enough to cause the disease using the similar concept of Mendelian genetics, whereby when you have big A, small a, it will still show red color. Okay, so that's the concept behind it. All right, all right. Phenylketonuria. Phenylketonuria, commonly caused by mutation of chromosome number 12. Okay, in this enzyme, phenylalanine hydrolase gene that is uh, producing the phenylalanine hydrolase, which prevent the conversion of phenylalanine into tyrosine. So one amino acid convert to another amino acid, it use phenylalanine hydrolase. So it just remove OH group from the phenylalanine, it become a tyrosine. So phenylalanine is essential amino acid, but excess is harmful. So you cannot have too much of this phenylalanine. So usually we are normal because this phenylalanine are converted to tyrosine by this enzyme. Okay, so some people are unable to produce phenylalanine hydrolase because the gene is defective. So that will cause phenylketonuric. So some symptoms result from excess of phenylalanine because unable to be converted into tyrosine, and other result from inability to make tyrosine. So sometimes in some people, because phenylalanine cannot be converted into tyrosine, the accumulation of phenylalanine is the one that cause the symptom. And in some other people, actually, the absence of tyrosine is the one that causes symptom. So that is why it is pleiotropic, meaning to say that from the same source of defect, different people behave differently, abnormally. Okay, so those with phenylketonuric, they have because these are the defect in the gene. So your entire cell in your entire body, right, have the same DNA, right, have the same DNA, so you cannot change anything. So the only way to treat this kind of defect or disease is by controlling certain things. So for example, in this case, accumulation of phenylalanine cause phenylketonuria. So you have to make sure that your phenylalanine amino acid in the individual with the defective gene has to be controlled. Okay, so diet is used to manage the phenylketonuric by providing just enough phenylalanine for protein synthesis our cells still need uh, phenylalanine to form all those amino acid to become protein. Our hair, our other enzymes for reaction. Okay, but then they cannot have uh, extra. That extra is the one that will cause the disease. So you just have the individual has to just eat uh, just enough in order for the cell to use, and then cannot eat excessively. Okay, by just providing enough phenylalanine for protein synthesis, but not enough that it accumulate when and cause the disease. Okay, and then this one actually is a side note. It's not really just to inform or one of the effect that can be seen is that NutraSweet is an aspartame. Okay, aspartame is a substituted sugar, fake sugar, for example, for those with diabetic, they cannot take sugar, right? Because they are diabetic. So, but still want to eat something that tastes sweet, coffee, sweet coffee. But if you eat the normal sugar coffee, you have cause diabetic. So, still want to have that sweet coffee. So, it take coffee with aspartame. But if that diabetic person is also phenylketonuric, then it will be a problem because the aspartame chemical compound, when it is broken down in our cell, then uh, it will break into phenylalanine. So aspartic acid is found in aspartame, but they modified a little bit so that those modification allow for that chemical to taste sweet. Okay, so when that happen, it becomes aspartame. But this aspartame has similar characteristic to aspartic acid. So when it is broken down, it becomes phenylalanine. So in phenylketonuric, it is it will be problematic. All right. Okay. So next time when you go to the supermarket or sometimes you go to a restaurant that serve coffee with a sugar in a sachet, and then sometimes you have additional one NutraSweet. You know, so you can read there at the back of the that sugar substitute. It will say that this not suitable for phenylketonuric. So you now you know because of the inability to convert this into tyrosine. Okay, when you eat this. Okay, but for normal people, we can take this because it will be converted normally into tyrosine by our cell. Okay, so this phenylalanine tyrosine metabolism is quite important in human in eukaryotic system cycle, especially you can see in human because this phenylalanine tyrosine and then the pathway related to it, you can see that if uh, from one amino acid, sometimes you require several different enzymes to reach certain end product. So during the reaction, if there is some deficiency in a certain area, it will cause certain diseases. But this one is the common one. So if from phenylalanine to tyrosine, if the enzyme is defective, cannot be converted, then it cause phenylketonuric. And then, but if in some other individual, phenylalanine to tyrosine can be converted uh, normally, but in some other individual, because it's the uh, inability, okay, to convert tyrosine into uh, dihydroxyphenylalanine, then that individual uh, will then cannot use this one to produce melanin pigment, then that individual suffer from albinism. Okay, so quite close pathways that can cause several different disorder. Okay, and then homogentic acid you come across before, if inability to convert this HA into malonyl acetoacetic acid, then it'll cause alkaptonuria. Okay, so these are a single enzyme or single gene disorder that can cause different a single gene disorder that can cause disorder in human. Okay, single gene over here cause this alkaptonuria. In another individual, single gene defective cannot convert, then cause phenylketonuric. In other individual, inability to convert tyrosine to DOPA, and that individual will be having albinism, for example. Okay, so albinism. So these are the further explanation. I'm going to skip that. Okay, Lesch-Nyhan syndrome, recessive mutation. Okay, so you look at this recessive mutation. What did I tell you earlier just now? If recessive mutation that cause the Lesch-Nyhan syndrome, it means that only those individual with both chromosome have the defective gene for this HDPRT that will cause the Lesch-Nyhan syndrome. So recessive means that both of the chromosome has to be defective. Okay, fatal disease is found in male. Okay, why fatal disease in male? Because male have one X and then one Y. So this one is X, this one is Y, right? So in male, only one chromosome X. So if the locus for Lesch-Nyhan is positioned over here, so if this one is defective, one copy is already uh, make them defective. So there's no other compensation. But in a female individual, oh sorry, in a female individual, it is two x's, right? X and X. So if this one is defective, but this one is okay, then it will be normal, isn't it? So that is why for those diseases or disorder that is related to the X chromosome or X link, okay, then you want to determine, although you heard the word recessive, you have to go another step to uh, to evaluate whether it will cause the disease in that person or not by looking at whether it is a male or male or female. So if a recessive mutation on X chromosome in a female, then if the female is heterozygous, then the female will be normal, right? But if the female is hemizygous, sorry, if a male is hemizygous like this, only one copy defective, then this one will cause the disease or disorder, even though only one copy is defective, but there's no another one. Okay, so that is the defect that can cause Lesch-Nyhan syndrome. Okay, and then another disease, Tay-Sachs disease, uh, called lysosomal storage diseases caused by a recessive mutation. Okay, you can see here the word recessive. So now you see whether the recessive here refers to autosome or X-linked. Okay, but this one is an autosomal, isn't it? So when it is autosomal, then you need both in to be in a hetero homozygous recessive in order to cause the disease. All right. So this one is the explanation and process ganglioside ganglioside accumulate in the brain cell cause some clinical symptom because it cannot be converted into other product to be removed in from the brain. Okay, so the disease is incurable. So most of the genetic disease it is incurable because you cannot change uh, the genetic constituent of our cell throughout our body. Okay, but then sometimes there is a possibility. Okay, for example, like if the disease is caused by certain enzyme, and that enzyme only function in the lung, for example, and then you do a gene therapy whereby that gene, the corrected version of that DNA for that enzyme is inserted into the lung cells. Okay, and then the lung cells then might be able to produce the corrected version to compensate for the original defective version in the lung. Okay, so that one is the concept behind gene therapy to treat certain disease. Okay, so this one is further explanation to the Tay Sachs disease, whereby the inability to convert into the product for the next reaction, it stop over here. So accumulation of this, it cause the Tay Sachs disease. All right. Okay, and then the disease can also, uh, you remember the DNA, on our chromosome can produce an enzyme, and it can produce protein. Okay, so protein can also, you learn in biochemistry, it can also become a structure. Okay, a structure which consists of beta helix and uh, beta helix and pleated sheet. Okay, so protein, uh, gene can also produce a protein structure, or proteins that function as a structure. Okay, so structural proteins, so such as hemoglobin, are often abundant, making them easier to isolate and purify. So defective gene can also cause a defective protein, and defective protein, because it cannot make a proper structure, so it cannot function properly, it causes the disease. So that can also happen. So it is not the gene don't produce the enzyme, but it produce protein, but because it is defective protein structure, then it cannot function normally. Okay, so that is one example. And uh, so defective gene cause defective protein. Okay, in addition to defective gene, cause or produce defective enzyme. All right, sickle cell anemia. First describe sickle cell anemia, finding that the red blood cell, through the study using the microscope, for example, uh, the shape of the red blood cell normal compared to the abnormal. Okay, so 1910, just a little bit later than Mendelian genetics. Okay, but then not as early as knowing that this one also caused by the DNA, because in 1950s, 60s only, then DNA was uh, fully described. Okay, but at that time they know already low oxygen tension. No, sorry, uh, low, not low oxygen tension. The different shape cause the inability for the red blood cell to hold the oxygen. So under, and then and then they find out that the red blood cell with in a situation whereby uh, the oxygen is less, then you can see the difference in the shape. So the red blood cell in the sickle form is fragile, hence the anemia. Okay, less flexible than normal red blood cell. And then those suffering from this, the effects are, you see, pleiotropic. So many different symptoms, and not all are shown in the same individual. Okay, so pleiotropic. So different individuals show different characteristic. And then a heterozygous individual will have a um, characteristic of this. Okay, but then at a milder form, because they can also produce the normal red blood cell. Okay, uh, so So in the 1949, they find out that they were result of a single mutant allele from the mapping. So from the mapping, because in 1949, uh, they know, for example, in Thomas Hunt Morgan, they know that chromosome is related. You still recall chromosomal theory of inheritance? Chromosome can uh, change in certain position on the chromosome, they can cause disease. So like this, they somehow able to map, and then they show that a single position changes that can cause sickle cell anemia. Okay, and then Linus Pauling and co-workers use electrophoresis. So electrophoresis, the concept is similar like the DNA electrophoresis, but in this case, they put the protein inside it. Instead of on the gel there's a well over here. Instead of putting the DNA and it migrate downwards, okay, they put protein inside it. Okay, so this one is also the electrophoresis of the protein. But sometimes and nowadays, usually in the gel for protein, the content or the constituent of the gel is different compared to the gel that you use for DNA electrophoresis. So protein electrophoresis and DNA electrophoresis, the concept is the same, but the constituent or the content of the gel in the electrophoresis are different between the DNA and protein. So when you take the protein from the normal individual and protein from the sickle cell individual, put on the electrophoresis and then give electrical charge and then let it run downwards, and then you can see that the way it runs, and then when you visualize it, they are slightly different. Okay, so individual from sickle cell trait show equal amounts of hemoglobin uh, A and S, okay, from the sickle cell trait, indicating that heterozygotes make both form of hemoglobin. So sickle cell mutation changes the form of its corresponding protein, and the protein structure is, of course, controlled by genes. Later on, it was found. So in this case, Linus Pauling found that in the normal individual, okay, in the normal individual, when you extract the red blood cell and then run on the electrophoresis, it form only one band. Okay. In some individual that show slightly symptomatic sickle cell, and when they uh, look at the um, microscope to see the shape, they found a mixture of normal and sickle. Then they see a mixture of the normal protein hemoglobin and slightly different hemoglobin. But those who are suffering from full symptom of the sickle cell anemic individual, when they run, you can see the position is different over here. So those in between have the mixture of the normal and also the sickle cell protein. So you can see over here, there is a difference because of how the protein are produced to form the hemoglobin structure. Okay, so hemoglobin structure is formed by four polypeptide chains, two molecules of each polypeptide, the alpha, and then two molecules of the beta polypeptide, each associated with a heme group. Okay, heme group uh, that can link to those four polypeptide. So, so hemoglobin actually is a what you call tetra. Okay, tetramer. Okay, tetramer because it has one polypeptide, two polypeptide, three polypeptide, four. Okay, one, two, three, four polypeptide. Okay, so it's a tetramer. Okay, so that is the the hemoglobin protein. So based on this, how many genes do you think is required to produce this tetramer that form the hemoglobin structure? Right? Write in the chat, please. Or you can say it aloud. It's also fine. Anyone? So far, no. Oh, okay, okay, okay. Wait, yeah, sorry. Okay. I didn't scroll down just now, so I missed some of the question. 251, 305. Okay, nobody has answered. So how many genes? How many genes required to form the tetramer hemoglobin structure? Come on. Oh, four. Okay, one answer is four, and then someone answered two. Four, two, four, two, two, four. So more or less, the idea is either two or four. Oh, there's one answer one. Question. Okay, one as the answer, and another one four or more. Okay, all right. Keep that answer in mind. No need to type anymore. Thank you. I will give explanation afterwards. Okay, but just want to address the question that someone asked earlier. Why do hemoglobin S able to travel further than the normal hemoglobin? A very good question. Why do hemoglobin S able to travel further than the hemoglobin A? Hemoglobin S is a sickle cell hemoglobin, slightly different shape compared to the normal hemoglobin due to a mutation of the gene that cause hemoglobin S. Okay, so when that happen, when you run on the gel, because the shape is slightly different, the charge of the protein, you recall your STB1083 biochemistry, right? Between alanine and another amino acid. Sometimes it has a higher or stronger positively charged molecule. So when that happens, it's slightly bigger than the other. So for hemoglobin S, the shape is slightly smaller by very small differences, but because of slight difference it moves faster, so it reach down earlier to the bottom of the gel. Okay, so that is why it travel further than the normal hemoglobin, because of the change in the shape of the protein itself. All right. Okay, so coming back to the question, so another one. Does the thickness of the line represent anything of the PCR? This one go back to the PCR. Usually in the PCR or in the protein, uh, the position first, yeah. In a PCR, if this is a gel, okay, if it's on the same position, usually it refers to the same size, whether it's a protein or whether it is a DNA that is being analyzed. Okay, but then uh, the thickness um of the band itself usually refers to how much you put your sample on the electrophoresis gel. Okay, you know when you run on the electrophoresis, okay, when you run on the electrophoresis, right? There is a, a well. You call it a well because it is like a hole where you put your sample in. So the thickness, if all the sample, assuming that they are the same size of the DNA, when you put a lot, then a big blob will travel, but they will be at the position. But if you put a little sample, and when you run, then you see very thin band. Okay, so so the thickness usually represent the amount of sample you put in. Okay, okay. based on this tetramer, how many genes required? So this beta polypeptide, this one is beta polypeptide. So this comes from same position. Alpha polypeptide, okay, on the same, assuming that they are on the same chromosome. This one, the alpha from here, the beta from here. So, so the beta will produce many different beta, but two are required to form here. So, but they are all from the same gene. Okay, so you need one gene for alpha, one gene for beta, so two genes. How many genes are required? Okay, so that's a concept. So you have to also recall, not only for this subject, is that when you have a gene that produce protein, sometimes the protein can exist in a tetramer like this, okay, but they uh, come from the same gene, okay, but and then they produce protein, same protein, okay, but when they want to function, they need six of the same protein, for example, okay, six of them to produce one functional protein. So this one is hexamer, okay, hexamer, and in some other protein, it requires four of the same. So uh, to form a normal protein function. So this one is tetramer, something like this. So tetramer can be, tetramer means how many subunit of the polypeptide. Okay, and then in some other instances, the tetramer will be two polypeptide mixed with different two polypeptide form a functional protein or functional enzyme of functional structure protein like this one. So this tetramer, this one is also a tetramer, but uh, they consist of different way of mixing of the polypeptide to form a functional protein. So that one is a chemistry, biochemistry punya subject. Okay, but uh, so it is not rigid like if you mention tetramer, it has to be 2 plus 2. It can be all four the same, or hexamer all six, or sometimes you can have three of different polypeptides. So it depending, like this one comes from two different genes. This one from the same gene, but the protein, the polypeptide are assembled. Okay, so have to bear that in mind as well. All right. So Ingram found the sixth amino acid of the beta chain individual is different. So from glutamine to becomes valine. So you can see over here, valine no electrical charge, whereas glutamate or glutamic acid in the normal is negatively charged. When that happen, the shape, although you see the position, the number of amino acid is the same. This one until blah blah blah, they are the same. But then, due to charges, then uh, it can uh, move a little bit faster. So this one move a little bit faster due to no charge, whereas this one is negatively charged, so it moves slower. Okay, and then in some other instances, when you have a mutant, you have a shortened version of the protein. That one you can also see the difference in the position. Okay, so also referring back to the earlier question, the changes in the charge due to the mutation that cause the changes in the amino acid will also influence the uh, how fast it runs in the electrophoresis. But but commonly it is because of the differences in the size of the amino acid. All right. So this one is further explanation. Uh, genetic description. The wild type beta chain allele is beta A, co-dominant with beta S. So co-dominant. So you can see over here, you have probably you have come across the word co-dominant. codominance with meaning to say that you coexist. Like your school, when you were in your school, you went to a co-ed school, right? And then another friend went to, for example, uh, apa a convent Bukit Nanas, all girls school, or last time, even now, MCKK, all boys, or Star, all boys. So that one is not a co-ed school. SK or SMK Gapo, Sekolah Menengah Sains Kuching Utara, Sekolah Menengah Sains Kuching is a co-ed school because uh, both male and girls and boys coexist, so co-dominant. So co-dominant means coexist. So they are not dominant over the other, right? So so uh, hemoglobin BA individuals have normal subunit, right? This one is a diploidic form, whereas in a diploidic form of the homozygous recessive, then has a bit the sickle subunit both with a low oxygen tension, so inability for the for the oxygen to attach to this heme group, okay, to this heme group. So oxygen not able to attach because the shape is slightly changes compared to the normal hemoglobin. Okay, so screening of hemoglobin for altered electrophoretic mobility has identified over 200 hemoglobin mutants. Okay, 200 hemoglobin mutants showing a variety of amino acid substitution in both the alpha and the beta chain. So each appears to derive from a single amino acid change, and most effects are not as severe as those seen in a sickle cell anemia. So basically, sometimes when an individual showing a slight symptom of anemic, but not a full-blown sickle cell anemic person, right? So different individuals that showing similar characteristic when blood are drawn from them, and then isolated the red blood cell and run on the gel, then you can see like the earlier electrophoresis, the normal one will be usually migrate a bit slower, but then you can see in the some slightly anemic, you can see slight changes in the position. Okay, not as a full-blown sickle cell anemic individual at the bottom here. So they see why this individual is slightly different. So then it is quite interesting because this one, they are not as normal as the normal individual supposed to be over here, but then slightly downwards. So there must be proteins, there must be some changes in their hemoglobin. Then you can start to investigate. Okay, now let's say we draw, take these people's DNA and see, and then when analysis is done, then they find that uh, gene different, slightly different, and then when they analyze the protein, amino acid slightly different protein uh, can cause slightly anemic individuals. Okay, so you can see like in the alpha chain, the normal will have this sequence. Usually, the changes that was found at this position number two, number 16, 30, 57, and so on, and then these were found in sample from Honolulu, Hawaii, then Virginia, Massachusetts, and in Philadelphia, for example. So different uh, location um, that have slightly symptomatic anemic problem. Okay, and then on the beta chain, some changes in this position. Different individuals have uh, different anemic and different amino acid changes. And when analysis is done, these are the changes in the amino acid from samples that were obtained in different places. Okay, this one can be in the US or it can be in in America, this one is in Canada, this one is in India, for example. Okay, so remember, uh, uh, changes in the gene, if at a single position, can cause disease. Changes in the gene at a single position can also cause changes to the protein that then become the disorder. Okay, so change in the structure or change in the enzyme function, inability to produce a normal enzyme. Okay, and that is only at one position. So these are example of disorder or mutants or mutation that can cause disorder or disease. Another example is cystic fibrosis, affects the cell that produce mucus. Okay, mucus. Usually lungs, digestive tracts, and then vast differences in the production of sperm. Okay, and and then in pancreas. So in any uh, organs and tissue that are related to the production of mucus, then it will cause cystic fibrosis, and usually it is involved the lung. Okay, characterized by abnormally viscous secreted mucus and lung complication managed by percussion and antibiotics to treat infection. And usually they produce a lot of mucus because of the inability of the cell to transport the mucus. And when that happens, the individuals will be infected easier compared to the normal individual. Just like when you have a flu, when you have flu and then you can have bacterial infect your lungs. So that bacterial is the one uh, that can live or strive in the mucus environment when you have flu, for example. Okay, so mucus um, secretion of the mucus secretion increase, and when you have that, lung complication happen, and that is because of the Q arm of chromosome 7 for this protein called cystic fibrosis. So this one again is another example of gene that can cause defective structure, uh, defective protein structure which cause the disorder. So in this case, you cannot see under the microscope like a hemoglobin because it is a very, very small protein that situated in between the membrane. Okay, so they investigated and find that they uh, actually they do the analysis and they found the gene that has similarity or homology with a family of active transport membrane protein. Our cells, when the cells need to transport something from one position to another. Okay, so when that happen, they cannot penetrate the lipid bilayer, the membrane, so they need to go through certain pores like this. So this one is actually pore. Sorry, this one is actually a pore that can allow certain molecule to pass through. Okay, to pass through this. So when that happen, so the mucus molecule come over here, and then it cannot pass through, so accumulation, for example. Okay, so that is oh, the description over here. So the defect, okay, commonly is a deletion. Deletion meaning to say that certain gene are, are modified by being removing one amino acid from the protein at the 3 base pair region. Okay, the 3 base pair region means that somewhere at the from the at the C terminus. Okay, so terminus, this one N terminus. So you can see that this one N terminus, C terminus, very long protein. Okay, it goes here and then over here, and then it comes over here. So between this and this, slightly longer over here, and then here, why so much? Okay, and then the defective position is over here. Okay, so most common site of the cystic fibrosis mutation, which is a deletion. So this the delta is a deletion of phenylalanine at amino acid number 508. Okay, of the entire transmembrane regulator protein. All right, so that is one of the um, cause of the disorder due to changes in the protein structure of a molecule. All right, so okay, then just a little bit genetic counseling. So genetic counseling is, sometimes when you are unable to test, so uh, when the test unable to be done, for example, you can do the analysis earlier by genetic counseling. So one of the way of doing genetic counseling is by doing pedigree analysis. Usually, this one involve of human. Okay, for example, the baby wasn't born yet, for example. So you cannot test on the baby still. Not in the baby is not full term yet, so you cannot do the analysis. So you can do genetic counseling by pedigree analysis, for example, uh, see the family history. Okay, so if this is the family history, the two individuals, these are their family history, got certain disease. This is the family history, got certain symptomatic similar disease. So based on the history of both two different families. So the daughter and the son from these two families, they want to get married. So when consulting the doctor, then the doctor will be able to advise. So what will happen? What is the percentage of having a defective embryo, for example. So that is done by pedigree analysis, and usually this one uh, done based on several generations history. Okay, usually it is done by to identify suspected carriers. So it's not so much of determining what's going to happen, but just want to find out if there is a possibility of certain person is a carrier for the disease, although that person is a normal individual. All right, then sometimes fetal analysis can be done by looking at certain enzyme or protein level. Uh, Sometimes you can also detect the changes or the DNA itself. Okay, for example, like spina bifida. Sometimes it can be detected when analysis is done through fetal analysis before the pregnancy come to full term, for example. Okay, most effective analysis there is currently no way to change the resulting phenotype. So genetic counseling focuses primarily on informing clients of the risk and probability. So this one, for example, if those get married and then the firstborn got some disorder, so consult the doctor, the specialist, and what is the percentage if the sibling, the couple, the partner pregnant again, and then what is the percentage of having disorder for the second children. So that is the advice that you call genetic counseling, for example. All right. And then a carrier detection. A carrier is heterozygous for a recessive gene mutation. So you can see over here a gene, uh, recessive gene that cause recessive disorder. So a person in the big A small a, for example, So the small a cause the disease, but if the individual is and big A small a, then this individual is considered as a normal, right? So when the normal carrying the recessive gene. So this one is heterozygous for recessive gene mutation. So carrier detection. In a cross between two carrier parents, one-fourth of the offspring are expected to develop a disease, and half will also be a carrier. Okay, because half by half, mother and father become 1/4. Okay, expected to develop a disease by the two parent of a carrier. Okay. Okay, and then carrier phenotype is normal, as I mentioned over here, is normal because the big one, the big letter is producing the normal enzyme to compensate or to cover for the one that is that is uh, not producing the normal one. So genetic counseling is also concerned with whether fetus is abnormal. So all of this basically just to mention to you that uh, the genetic disorder that is caused by changes or mutation in a single chromosome that, like I have mentioned earlier, uh, can be detected by genetic counseling, can be predicted by genetic counseling, and can also be detected by fetal analysis. Okay, so these are how the genetic disorder is nowadays being detected. And also I have to say that for fetal analysis, the analysis is not only for disease that is caused by single mutation of single gene that cause a disorder, but fetal analysis can be used for other type of diseases that is caused by multiple enzyme disorder as well. Okay, so that will be the end of our lecture today. So we finished quite early this week. All right. So anything that you would like to ask in the chat box? There is no question as yet. Again, if you don't want to ask now, you can also ask later in the forum that I have created. Okay, so far I have one. I have yet to put the copy the answer that someone asked me. I think I have uh, collected the question. Okay, can you brief about quiz one? It's already inside there. Two questions only. Okay, this lecture is recorded. Uh, does quiz lecture one will be counted as a carry mark? I hope I want. I actually I wish. No, actually I wish. I want to use it as a carry mark because it's two questions only. All right. So don't worry. You know when I do a question for quiz anything, you don't worry that oh dear, Dr. Hasnan is doing a quiz, so I need to do a revision. You just do your normal revision, right? You just do a normal revision, and then just attempt the quiz. You remember, I will do several quizzes, and then and those quizzes, usually I will take into account for the 5% after I average them. Okay, and then the question, like the first one is two questions. Probably the following quiz after I finish this lecture, there will be a follow up of another quiz, quiz number two, probably five questions. Not that many. It's not like you have to do some 20 questions. This is MCQ question. Okay. So don't worry. Don't be too stressed about it. All right. If you cannot answer, then that is fine. Don't, you read later, and then so you learn something from it. Uh, don't don't really, basically don't worry too much lah. Kan tak perlu risau sangat lah. I mentioned about quiz then kamu dah panic lah. Right. So I think that's all. Any other question? All right. Um, this is another thing, another question that someone asked me. I don't know. Let's say before I mention that, let me see how many of you today. 119. Okay, I know that there is there are another 119, 20, 130 of you, right? So there are 10 students who are unable to join, and there will not be penalty for them who are unable to join. Okay, so uh, I um, I have to emphasize that because I know that this is a lecture that's supposed to be for group number three, only 30 something. Okay, but then uh, I have to thank you as well because you are the other groups are able to join. So those who are unable to join, I know that you or those who are unable to join, they might have their elective courses or something like that. Okay, so I assume they have elective courses and unable to join, so they can always get the recorded lecture uh, and learn from there, and then they can always ask question later on, right? So I think at the moment I haven't decided yet on on determining whether, okay, what happened to this? Can you see that? Okay. Whether it's going to be Thursday still, or I want to do on another day. Tell you what I can do is that I will just go for I will do a quiz, not quiz, a survey, a simple question. Will you agree to continue the lecture on Thursday 2 to 4? If you don't agree, then probably the next question, uh, uh, when is the most suitable one, and I'll decide based on that. I'm supposed to do that quiz last week, isn't it? But I haven't done that, so I just go ahead and do it. Okay. So any other question from any of you? Um. Okay. Manual attendance. Okay. I'll check afterward. At the moment, I haven't decided. Zach asked me. I haven't decided, but based on the number, I'm inclined to do on Thursday. But then, never mind. I will hear your opinion first. All right. So, just go to the elite pages and then have a look there for announcement. All right. For announcement, I will put it there. Okay. For Tuesday next week. All right. Okay, like this. For Thursday, for next week, sorry. For next week, we still do Thursday because I'm supposed to fly back on very early morning Tuesday. So I don't know whether if we decide to do on Tuesday, definitely 8 to 10 is out of question. 10 to 12 I might make it. So so let's say for next week we decide now, we'll do Thursday. All right, let me see if Thursday got any holiday or not. Deepavali, so how many oh dear, Deepavali is a holiday. uh, Probably some of you might want to go for a holiday, for um public holiday, isn't it? Not fair for me to do a lecture on Thursday. Okay, okay. Wait for the announcement lah. At the moment, tentatively on Thursday, and then I will find out further whether Deepavali is a public holiday or not in Sarawak, because I know in school Thursday is not a public holiday. But then in the Unimas Almanac, it is bold in the letter, so it might be a public holiday. I'll find out from there. Okay, okay. I'll confirm it in the elite. All right, so that's all. Thank you very much. You might have other things to do. So that's it for today. Thank you very much for being here. If you have question, please ask. All right, please ask. If I cannot answer immediately, I will attend to that a bit later. If I still don't answer, remind me. Jangan takut. You know, my job is not to school all of you. My job is just to make sure that you learn something from it. Okay? So that's all for today. You may leave. Thank you very much. Stay safe. Hope to see you all again.
Thank you, Doctor.
Thank you,
Doctor.
Thank you,
Thank you.
Doctor.
Okay, bye-bye.
Doctor.
Thank you,
Thank you,
Doctor.
Doctor.
Welcome.
Have a nice
You're
day.
welcome. You too. Bye-bye.